It's a pleasure to be here. And, okay. And we're on. Okay. Thanks very much. This one. Backwards, this one. Okay. Terrific. Okay. So basically, ladies and gentlemen, this is what we would like to be in this situation. Or at least I would like to have the capabilities to engineer mini submarines. Mini submarines that will float inside the blood vessels, identify diseased cells, target them in a very specific and safe manner, deliver drugs or manipulate them, eradicate them, or change their expression level of genes. This is the dream. Can we make it a reality? So basically already, and this is from quite an old review that we published in Nature Nanotechnology in 2007, basically you can see here just as examples, but there are almost more than 35 nanodrugs already in clinical use. With the first one was Doxil, Xilex actually was mentioned today, a liposome-based formulation that was approved by the FDA in 1995. So if we look at clinical trials today, we have more than 350 clinical trials in different types of nanocarriers, mainly for cancer, but recently also for inflammation and viral infection, which this is very, very encouraging. So, okay. Wow. In a recent, very, very recent, last week actually, perspective that we published, we looked at the area of personalized medicine. And here we suggest that not only the drugs that are tailor-made for the particular patient should be designed, but also the drug carriers with it. The laboratory of nanomedicine at Tel Aviv University was established almost 3.5 years ago when I accepted the position after being in Harvard for a few years. And the lab has two arms. It's quite unique. An arm for drug delivery and targeting, which we actually make nanoparticles from different shape and sizes and surface modify them, but they're all made from naturally occurring biopolymers, sugars, lipids, and proteins. And we are part of the immunology department, so it's, for us it's very unique and important that they will be very safe. So we have a very special nanotoxicity or immunonanotoxicity program in our lab. We also design and engineer proteins to make targeting agent in a very selective manner. The other part of the lab is doing much more of biology, doing target discovery and validation, usually with RNA interference, but also with small molecules, libraries, and peptides. And here we have a high throughput facility, and actually we can basically find new drug targets. And the ideal situation is to try to combine this arm with this arm and to create a personalized approach, again, tailor-made, the appropriate vehicle, the selective targeting agent, and the specific therapeutic payloads. And all goes into translational studies and hopefully into the clinics. So the lab is mainly a focus on leukocytes-implicated diseases, and this is a huge field that includes blood cancer, inflammation, and viral infection, and we are quite occupied in each of those buckets, and I'll try to share with you a little bit of the data. So we are building on two types of innovation, RNA interference therapy that can expand the therapeutic space and streamline drug development, and today, from the time we actually have a target in hands and into the clinic, it actually can be 15 months. So very, very short time. The only problem is how to deliver those payloads. So from the nanotechnology standpoint, we're trying to engineer nanocarriers, carrying high payloads of those molecules, having a selective specificity into specific cells. We want to have healthy cells untouched. Very difficult to have, but we're trying. And we want to have a safe design of those carriers. Okay, so this is a model cell. And this, I found it in a very scientific website called Google, and you can see that if this is the cell, this is the nucleus, you have a DNA inside, and it will be transcribed to a messenger RNA, the ribosome will be hitchhiking here and basically translate to a protein, a 3D structure, and this is the beauty machinery of life. The discovery of RNA interference in 1998 and the Nobel Prize that was awarded in 2006 opened new avenues in this area. Basically, now you can design a sequence-specific drug that can eliminate the messenger. There is not going to be translation, and at the end there's not going to be faulty protein. And just imagine how pharmaceutical companies looked at this when it all started. Potentially you have now the ability to create any drug druggable, any target druggable. However, it's not so simple. So I'm going to actually share with you some biological stories. And the first one is about cyclin D1. This is a cell cycle regulating molecule that governs the proliferation of both normal and malignant cells. Therefore, it's a known target for cancer. However, in inflammatory bowel diseases such as Crohn's disease, ulcerative colitis, chronic pouchitis, cyclin D1 is aberrantly upregulated, both in the epithelial cells and in immune cells. Now, its role in epithelial cells have been well documented. It's connected to cancer, carcinogenesis. The role in leukocytes, who knows? About 5 years ago, we set up a strategy, very naively, to target gut leukocytes and selectively silence cyclin D1 in an experimental colitis model. Now, systemic RNAi delivery is very challenging, because we know that many cells, even cells that sample the environment, like dendritic cells and macrophage, do not uptake RNAi molecules without any transfection reagent. They are, in general, the molecules are degraded very fastly, and there is a fast renal elimination. However, in leukocytes, this is a much greater challenge, because leukocytes are intrinsically resistant to transfection by lipid mechanism. Even with viruses, primary leukocytes are not the easy target, and they have diverse distribution pattern, often located deep in tissue, so it's hard to target them. So we are looking for receptor targets for siRNA delivery into leukocytes, and there are actually many options. But one of the options we find very interesting in the leukocytes, integrins. Integrins in general are the largest family of cell adhesion molecules that mediate cell-to-cell and cell-to-matrix interaction. And here is a schematic illustration how leukocytes interact inside the blood vessels with the endothelial cells. And the importance of those receptors on the leukocytes and the epithelial cells is very important, and they are part of the integrin family. So we created a strategy that we called integrin targeted and stabilized nanoparticles, and actually I'm going to show you that we can expand it. And this was the first example of nanocarriers made from naturally occurring lipids, neutral in charge, coated with hyaluronic acid that we heard today, or HA, CD44 receptor. But here, it's actually a scaffold for putting an antibody on a surface and for lyophilization. We put an antibody or a fragment of antibody directed against the leukocyte integrins, and we can encapsulate siRNA, and all was documented. Just to give you an example what's going on in flow, so if we take beta-7 integrins that are actually expressed in the gut, and this is something I have to explain here, it's actually very important that when you have inflammation, some T cells, circulating T cells, home into the gut. And we try to utilize this as we believe that those leukocytes are the best drug delivery system. So if we can target the leukocytes in the circulation and we can penetrate into them, they can home to the pathological area and do their job. So instead of us being very sophisticated, we just hitchhike on what nature do best. And this is an example. We took splenocytes, mouse splenocytes, also expressing mild amount of beta-7 integrins, and splenocytes from a beta-7 knockout mouse. And we pre-labeled the antibody on the surface with a fluorescent dye, green one, Alexa 488. The siRNA payload was with Cy3, and this was done in a flow chamber. Basically, it's under flow. It has to be very, very fast reaction. If those particles are not binding and internalizing within 45 seconds, we have a deep, deep problem. So what you can see here that if we have an isotype control, nothing happens. You don't see any fluorescence. If we have siRNA alone, they will not penetrate into leukocytes. If we use this delivery system, you can see very nicely how we get co-localization. And if we use a knockout mouse, nothing happens. Very happy from this. We worked our way up, and I'm not going to talk about all the stuff, but one of the interesting discoveries, and it's a biological, very important phenomenon, excuse me, that when we silence in vivo cyclin D1, we found a very interesting phenomenon. Some of the pro-inflammatory cytokines went down as well. And it means that TNF-alpha, IL-12P40, and therefore also IL-23 are going down, and the anti-inflammatory remains the same. It means that potentially we have now the means, if this is true, to shift from a pro-inflammatory situation to an anti-inflammatory situation. And there are many, many types of diseases, inflammatory diseases, and this is exactly what you need. So it's not that you're going to mix hundreds of different drugs together, even not 3 or 4 antibodies, monoclonal. A single gene can control many things. How it operates, it's going to be a paper very soon coming because we now reveal the mechanism behind this, but it's a transcription, a co-transcription factor as well, cyclin D1. So just to see if we can target the gut, the beta-7, with a colitis model. And before that, I'm going to, do around and a bit shift here, because we wanted to use a micropet CT, and we actually published a nice paper 1.5 years ago, and it was highlighted also by Nature Drug Discovery. The idea was to take the antibody against beta-7, to put a chelator, to put copper-64, and to do an imaging. The imaging, basically, you can actually do now early detection for Crohn's colitis, because, as I mentioned, those leukocytes home without any help from the circulation into the gut. And what you can see here, this is a single IV injection, and you can see nicely that we can, 48 hours after a single injection, we can track about 7.5% of the injected dose into the gut area. Of course, you have non-specific uptake, but into the gut area very selectively. If we do it with an isotype control system, then we get neglectable amounts, less than 0.2% of the injected dose, and you can see that the liver is actually full of this reagent. This was licensed, and it's now in a phase 2 clinical trial by Takeda, who actually owned the antibody. And then just to mention, to finish up, wrapping up the story with the cyclin D1, so we totally believe this is a good target because we can block intestinal inflammation. You can see the histology here. This is what's happened in colitis. Infiltration of leukocyte destroyed the gut area. And here we get a little bit inflamed area, but it looks very promising. And everything else was documented in a Science paper 2008. Is it the platform? So basically we believe it is. We believe that by changing the antibody on the surface and changing the payload, you now be able to target different subsets of leukocytes. Another example, multiple myeloma. Okay, an incurable B-cell malignancy in the bone marrow. This is a PET scan, and all the black dots are metastases. So we decided to utilize this platform and see what we can do. And I'm not going to go into this because also this was published recently in Kim Society Review as a review, and before that in Nature Nano. Basically, we use the same strategy, but instead of using an integrin antibody, we use an anti-CD38 antibody that expresses in many B cells and also in myeloma cells and plasma cells. Okay, and just to make a long story short, we had a pilot study very recently in September 2011 in Dana-Farber with Ken Anderson as the investigator. This is actually the first targeted nanoparticles specifically into blood cancers. And it was approved. There are 10 patients. Everything looks very nicely. Blood counts, liver enzymes, nanotoxicity parameters, and it will be published very, very soon. So if it's a platform, then we can go and tackle other subsets of leukocytes. Another example, HIV. So in a work that we published 1 year ago, again, leukocytes-directed siRNA delivery, we can suppress HIV infection in humanized mouse model. I'm not going to go into the detail because this model was published in Cell in 2008, and we actually used the strategy to target leukocytes in this mouse model. It's actually the only mouse model available for HIV. Basically here we took an antibody against pan-leukocytes, against LFA1. We put either CD4 siRNAs or CCR5 siRNAs. And again, the antibodies and how we make them were published in a set of paper, 2 in PNAS, 1 in Nature, Nanotech. And just to show you some of the results that also were published, we can definitely at the protein level, after a single IV injection of 1 mg per kilogram body, see very nice protein reduction in the blood of CD4 cells, T cells, spleen, and liver. Almost clean results in CD8. And if we do a challenging experiment where we basically challenge the mouse, okay, and look for a total number of T cells and viral load, something wrong here. Anyhow, each line represents a mouse. You can see that there is no infection. This is the basal level. When you have an HIV infection, up to 55 days, we can actually see reduction in the total number of T cells, and we can, using siRNA against CCR5, we can protect from this in reduction of T cells. I just want to mention one very important thing is that those viruses are HIV-BAL. So the affinity for CCR5 is very, very high. However, quite surprisingly, in the viral load, this was the basal level, this is the jump as expected because it's a logarithmic scale. However, this is an unexpected result. Somehow, it probably means that they have other alternatives, or because it's a knockdown and not knockout, we still have enough CCR5 and the virus can actually multiple inside. Okay, so what are we doing these days? We actually are involved in many, many projects, but as I mentioned, majority or half of the lab are interested in making different particles from naturally occurring biopolymers. These are lipid-based, about 100 nanometers. These are completely sugar-based, okay? And so you have about 30, 35 nanometer in diameter. Looks like viruses, but actually we are mimicking envelopes of viruses without any lipids. So it's only sugar. I want to switch gear a little bit and tell you about something interesting that is for solid tumors. So the Gagomer system, it's a lipid drug cluster surrounded by hyaluronic acid. This also was published in a few papers last year and this year. Basically, it's a cluster of particles, and this cluster of particles can either entrap small molecules or RNAi in different formulations. But what I'm going to show you here, that the ability to entrap, for example, paclitaxel, an insoluble drug, is amazingly much better than any formulation that we know. So if we look at the pharmacokinetic in mouse in the CD26 tumor models, again, this is paclitaxel in a chromophore formulation, very fast plasma clearance. Here we have up to 24 hours. If we look at the accumulation in the tumor is about tenfold with this system. We have reduced spleen and liver uptake, which is dramatic. And if we go to other parameters such as tumor volume, it looks as good as injecting fourfold higher dose of paclitaxel chromophore formulation. And if we even look and do a head-to-head comparison with Abraxane, this is exactly what's happening. So this is the Abraxane. We don't see any therapeutic benefit, although it's much safer for the patient as well, because it has no chromophore inside. And in this system, we don't have chromophore as well. It can actually solubilize 2 molecules for every molecule of the delivery system. So for the raw material in the delivery system. So it's really amazing. And it has the ability to distinguish between tumors, cells, and non-tumor. And this was published a few months ago from the Department of Head and Neck Surgery in Rabin Medical Center. They took head and neck tumors and they stained for CD44. CD44 expressed widely in many epithelial cells as well. And you can see here an example. The same patient has tumor on the one side, probably is a thyroid, and the other part of the thyroid is free from tumor. You can see that the antibody does not distinguish between those. Basically, it's a flow cytometry, so it binds very nicely to the tumor cells, which is green, and to the healthy cells from the same patient, which are with the red. However, the delivery system, if we label it with a FITC, with FITC, or with Alexa 488, can distinguish. And it means that there is a conformational issue here, an alternative splice variant, that only the natural ligand can identify, and the pan antibody cannot. Of course, if you use a specific antibody against this specific variant, in this case it's variant 4, it's not very known, you'll get the same results. So nature is more smart than we think, and it utilizes very sophisticated machineries to overcome many things. So just at the end, actually, I can talk a little bit about this. I have one or two slides. What drives us so much crazy about hair? So now you're going to laugh a little bit, because actually we are interested also in manipulating hair follicles. And this is not the only reason, right? It's not the only reason, yes. Actually, it's more sophisticated. Basal cell carcinoma and squamous cell carcinoma, some of them have been shown very elegantly to start with bulb stem cells, which are the stem cells of hair follicles. And we are developing a strategy, and I apologize here because of the MEC inversion. We are developing a very specific strategy, which is local, to target bulb stem cells, and we already have a panel of genes that if we manipulate that, you can change your color hair. This was published, and another one that actually can make your hair grow. The only problem with this, that we also induce cancer with this. So I'm actually giving the opportunity from the next speaker that will talk about toxicity to understand what I'm going to say. I want to thank the lab members and all the ones that are in balloons here, just weren't around, but this is on the beach of Tel Aviv, and it's about 5 minutes from the lab, so you can imagine that people want to do everything but science. And the funding agencies that help us a lot, and I want to thank you all for listening.